Interpreting Brief. A cell biologist researcher, Amir Herani, is presenting at an international cell migration conference in London on the results of his research. The audience consists of fellow cell biologists, cancer researchers, and medical professionals. You have been assigned this speaker and must interpret the speech using the simultaneous mode in a conference interpreting booth. The monologue task begins now.
Ladies and gentlemen, it is my great pleasure to be able to talk to you today about how cell migration in the nervous system works. Let me start by saying that although many neurons resemble other cells in developing embryos, in migrating long distances before they differentiate, neurons differ from other migrating cells despite sharing basic machinery. The most dramatic difference is that migrating neurons have a long and dynamic leading process and may extend an axon from a rear while they migrate. Neurons must also coordinate the extension and branching of their leading processes. Cell movement with axon specification and extension, switching between actin and microtubule motors, and attachment and recycling of diverse adhesion proteins. It is important to fully understand how migration of such morphologically complicated cells is coordinated over space and time. So, nervous systems are the organs through which animals perceive, interpret, and respond to the world around them. They consist of specialized electrically active cells connected together in networks. Essentially, all nervous systems develop by four main stages. The proliferation of progenitors in an epithelium, the specification of neurons in glia, the growth and guidance of axons and dendrites, and the development and refinement of electrical and chemical synapses. However, some more complex nervous systems, including those of the vertebrates, have another stage in which newly specified neurons migrate before they differentiate and form synapses. Some migrations cover long distances, up to thousands of cell diameters, and they follow complex routes, changing direction at landmarks along the way. So this ability to migrate means that neurons from different proliferative zones, correspondingly distinct lineages and genetic programs, are able to position close to each other and communicate, which might increase efficiency. In addition, different types of neurons arrive at a particular location at different times during development, so circuits are established in a specific order. This is why it is generally thought that neuron migrations facilitate circuit formation and improve nervous system function, although this hypothesis has not been critically tested by the appropriate mutation studies.